Good day to everybody. Today I am going to discuss about the lysogenic cycle of bacteriophage or virus. So today our topic of discussion is lysogenic cycle of lysogenic cycle of bacteriophage or virus. In my previous video, I already discussed about the lytic cycle of bacteriophage. Lytic cycle, there are two types of life cycles happened in bacteriophage or virus. One is the lytic cycle, which where the virus is called virulent, virus or bacteriophage is called virulent, and another is the lysogenic cycle. Lysogenic cycle where the virus or bacteriophage is called avirulent. Means it is virulent means it is infectious, it will kill the bacterial cell, lyse the bacterial cell, and in this case lysogenic cycle, the bacteriophage or virus will not kill the bacterial cell. Actually here the bacterial virus or bacteriophage DNA will be integrated into the host bacterial cell genome or DNA, chromosome. So this is called lysogenic cycle, and actually what happened, this when the virus or bacteriophage will be integrated in the host cell genome or chromosome, this is then this type of bacteriophage or virus is called virulent, sorry, temperate phage. And when this DNA will be inserted in the bacterial genome, this DNA virus genome or bacteriophage gene in the bacterial cell gene is called the prophage. So prophage, it is called prophage. And when this phage DNA will be integrated, it is called the temperate phage. Now, now the main thing is the when this in lysogenic cycle, when this viral DNA or bacteriophage DNA will be inserted in the bacterial DNA or genome, the bacteria will become resistant to lysis, means it will be not lysed by the phage DNA in the case of where it is happened in the case of lytic cycle, but here the bacterial cell will not die, and it will be not lysed by the bacteriophage or phage DNA, but and also it will become it will develop its immunity power, resistant power to the particular bacteriophage or phage which was attacked by the bacteriophage or phage, related phage. And here the adsorption will be happened like lytic cycle. The bacteriophage will inject its DNA into inside the bacterial cell, but here the and in lytic cycle what happened, the bacteriophage DNA will replicate by using the metabolic mechanism of bacterial cell, while in the case of lysogenic cycle, they will integrate it in the, they will not replicate the viral DNA or bacteriophage DNA. They will, their DNA will be integrated in the bacteria cell genome or DNA. So this is the thing, and how this is happened in genetical level, I will discuss about that, so that you can understand it better. Like that, when the actually when the lysogenic cycle will be happen, one gene, one protein like immunity repressor protein, which is produced by the immunity repressor gene of the bacteriophage. Actually it will stop, it will block the transcription and the translation of the expression of the gene, like in the gene expression in the prokaryotic cell, the lack operon. Whatever in the lack operon, there is a repressor protein which will bind to the operator region to stop the production of the beta galactosidase gene. Here same thing will be happen. Always the bacteriophage will produce the immunity repressor gene, which will protein which will block the production of other late gene. I have discussed about the phage DNA, the late gene. It will also present in the phage genome, the immunity repressor protein gene. Actually this they will stop the lytic cycle of the late gene of the lytic cycle, means the production of phage head, bacteriophage head, bacteriophage tail, bacteriophage fiber, and also the stop the production of the lysozyme enzyme, endolysis enzyme, which will help to lyse the help in the lysis of the bacterial cell, to kill the bacterial cell. So altogether all this gene will be, late gene will be stopped by this immunity repressor protein. And another protein, when the another this bacteriophage DNA will be integrated in the bacterial DNA. This is okay genome. Sometimes this bacteriophage DNA from which is present in the bacterial DNA by lysogenic cycle, it will be again, it will can be again released by some induction. Induction through some irradiation by ultraviolet rays, or the some exposure to the some chemicals, it will be happen again. The gene of bacteriophage will be released from the bacterial cell itself, and it will start the lytic cycle again. And this gene is this gene is the progene of bacteriophage, which will produce some protein suppressor, which will suppress the actual immunity repressor protein and start the release of the gene from the bacterial genome, and start the lysis cycle and kill the bacteria cell again. Start the, so this progene and the immunity repressor gene will be simultaneously responsible for the lysogenic cycle happening or not. Actually always the immunity repressor protein will be produced to stop the lytic cycle, but when the progene will produce the protein repressor gene, which will block the production of the immunity suppressor gene, and again start the lytic cycle, then the expression of other late gene will be started, and the lytic cycle of the same phage will be happen. So in this way, this two cycle, lytic cycle and lysogenic cycle will be happen. So there, and another thing is the induction of the lytic cycle again after lysogenic cycle happen due to the another gene. These are present in the bacteriophage gene. These are bacteriophage gene, which is present in the bacteriophage genome, and another gene is the REC-A gene, which will be produced by the actually this model of lysogenic cycle is studied in the lambda phage of E. coli, which will affect the E. coli bacteria. This lambda phage, it is with lambda phage of E. coli bacteria. And this gene is the, these are the lambda phage gene, progene and immunity repressor gene. but this gene, REC-A gene, is the E. coli gene. E. coli gene, which will, when the induction, this spontaneous induction, which is release the bacteriophage gene from the bacterial cell and again start the lytic cycle, this enzyme, this gene will be responsible of E. coli host cell, not from the phage genome. It is from the host cell genome, E. coli genome. It is the host cell E. coli which will infected by the lambda phage. This is the thing, and this will actually suppress or repress the or breaks down the immunity repressor protein and again start the lytic cycle. So REC-A gene of E. coli is responsible for the spontaneous induction or start of the start of the lytic cycle. So this is the thing, and there are some practical application of these things happen. Examples of these things: lysogenic cycle, like some pathogens like Corynebacterium diphtheria and the streptococci cause the scarlet fever, the erythrogenic production of some rash production in scarlet fever in case of streptococci bacteria, and the botulinum botulinum toxin is produced by the Clostridium botulinum, is only happen when there is some lysogenic cycle of bacteriophage will be happened in this pathogenic bacteria. This is the called the lysogenic conversion of the cell. This, when these bacterial pathogenic cells will only produce the this type of toxin when there will be lysogenic cycle present in their genome. So this is very important. And another thing is the I previously discussed that the conceptual model of oncogene, means some viral genes which will cause the cancer by this lysogenic cycle, like human papillomavirus, hepatitis virus, Epstein-Barr virus. This virus has the ability to integrate their DNA in the bacterial cell, in the host cell genome, like a human cell genome, and cause the cancer in the whatever the gene are responsible, apoptosis related genes are responsible, they will affect that and they will cause the mutation there. We know that. So this is the thing. And another bacteriophage is the mu virus of E. coli also. It is not. It is also infect the E. coli virus. It is mu. Actually, mu is a short form of mutant. Mu-phage. Mu-phage. It is called mu bacteriophage of E. coli. Actually, when this mu-phage has the ability to integrate their multiple copies of gene in the host genome and cause the mutation there. That's why it is called mu phage. And this is caused by the int gene of their phage genome. Int gene will cause this type of integration of this their gene in the host cell genome. Yeah, this is called int gene. Now, when the I have told about that the lysogenic cycle of bacteriophage or bacteriophage or the virus, there are this lysogenic cycle can be happen in when this is happened. This is happened due to two type of gene transfer methodology. One is the generalized generalized transduction, and another is the specialized or restricted transduction, restricted transduction. So what is generalized transaction? Try to understand what is transaction. Transaction is the called the transfer of gene through bacteriophage or virus. This is called transaction. And here there are two type of transaction: generalized transaction and restricted transaction. I have told that when lysogenic cycle will be again reverted to lytic cycle by some induction, like irradiation by ultraviolet rays or exposure to some chemicals, it will be happened. So when they are released from the gene of the phage gene will from released from the bacterial cell, what happened, bacterial genome, they will due to the sometimes due to the excision fault, so can't the phage DNA from the bacterial DNA, there will be some fault or error. So that what happened, sometimes this phage DNA will be released with some portion of bacterial DNA and they will be transferred. So this is called transduction, and it can be happen generally. What is generalized transduction? Generalized transduction when bacteriophage DNA, virus DNA will be integrated any place of the genome of the bacterial cell. This is called generalized transduction. It is happened in the case of P1 phage of E. coli. P1 phage of E. coli, and it is restricted transaction. Restricted transaction only happen at a particular specific phase of the bacterial genome. This is called specialized or restricted transaction. It is seen in the lambda phage of E. coli. This will be seen in lambda phage of E. coli. So what happens? They will release with some bacterial DNA. Phage DNA will release with some bacterial DNA to again start the lytic cycle. So they will take the bacterial DNA due to the excision repair, excision fault or excision methodology fault. This will be released with the phage DNA, the bacterial, some portion of bacterial DNA. Now, generalized transaction, what happened in the P1 phage? If you hear it, then you can understand it better. There are some genetic marker generalized transaction. There is some genetic marker is there, like genetic marker means some amino acid like 309 positive, like leucine positive, like azide positive. So I suppose that the E. coli cell, this E. coli cell is generally these are positive, 309 positive, leucine positive, and azide positive. Now it is infected by some bacteriophage. Now it is infected by some bacteriophage, and their lysogenic cycle by this bacteriophage, they will be integrated here, bacteriophage DNA. So what happened when they will be released this again to the lytic cycle, then this will take the adjacent portion of the bacterial gene here extra with them. So what happened, they may take the 309 leucine in azide gene with this bacteriophage gene, and they will again infect if we infect the another bacterial E. coli cell which was 309 negative, leucine negative, means they are not producing 309 leucine amino acid, and resistance, agide resistance positive, agide resistance positive, agide resistance negative. So what happened, this type of bacteriophage with the bacterial DNA again infect the this E. coli cell, then these E. coli cell are actually negative. They cannot produce threonine leucine and they are not resistant to sodium azide salt. So now they will become, again they will integrate this thing by lysogenic cycle, they will integrate there and they will be released by the spontaneous induction, and they will actually if the gene this gene will be integrated by the lysogenic cycle and they will start producing the threonine, leucine and azide, and they will become positive here. So we can determine this by the some genetic marker process, like we have to grow this E. coli cell in the medium devoid of this threonine and leucine, and they will grow at this time. This negative E. coli cell will grow means they will acquire the gene of this they are from this the phage DNA. Phage DNA will transfer this. This is the called the transaction, gene transfer. It is happened in the generalized transaction methodology, and for this type of thing will be useful for the genetic mapping of the bacterial DNA. What is the position of particular gene in the bacterial DNA, we can guess from there. How? Like that, this whole bacterial gene, this is bacterial DNA, E. coli DNA. Here the threonine will be there. Say that here leucine, threonine here, threonine gene, here leucine gene, and here the azide gene will be there. So sometimes what happen, when threonine positive gene will be transferred here in the bacteria cell, it has been seen that the only 3% will be leucine and no azide positive. Sometimes when threonine positive gene will be transferred, and when leucine positive transfer, then the 50% of the azide positive. Means what this statement will show that the actually the sequence of the gene is the threonine, leucine in the genome, azide positive, and azide positive is more close to the leucine gene than the threonine gene. So that's in this way we can genetic map of the any bacterial cell can be determined by this method. So, and this is the generalized method of generalized transaction method, and another transaction method is the restricted transaction method, which is happened in the lambda phage of E. coli cell. E. coli, this is a bacteria E. coli cell. Here they are specified, they are specifically bind to the at the specific position of the bacterial DNA, E. coli DNA, and this is the, they are between the gal or bio gene. This gene, generally it is written in the gene will be denoted by the small letter gal and bio gene. Gal means galactose utilization gene, metabolic gene, and bio means biotin production gene. Actually here biotinidase enzyme will be produced, which will bioavailable the biotin for our metabolic processes. So many amino acid, carbohydrate, this biotin is used. It is a vitamin B type vitamin, vitamin B, and it is generally useful for this different metabolic protein. It will be used as a coenzyme there. So here the lambda phage will be integrated in between gal and bio gene of E. coli. So when they are released from the E. coli genome, the lambda phage gene, they will take the gal or bio gene of the E. coli. So this is the for genetic engineering. These bacteriophage are very useful as a carrier, or you can say that as a carrier for the gene transfer in genetic engineering method. This bacteriophage and the plasmid will be used as a carrier for the gene transfer method. This is called the transaction. So today I think it is enough for today. So next time we will meet again.